Dr. Priya?
Can you hear me?
Yeah, of course. Are you okay?
In the sense what? I mean, I would like to. Can you?
Actually, we want to check whether the system is okay. Are you able to share the content? Something like that.
Right. Yeah. So where you want to see that?
You just try to share your content first. Click on. Can you see the control bar?
Yes, I do. Do you need to give me access?
Okay. Okay. Priya?
Yes.
You can share from there using the share button on the control bar.
There's the share tray.
It's not shared yet. Can you see the control bar over there? On the screen?
You mean the camera, mic, everything is right?
Yeah. Video, camera,
Right.
of course, red button for leaving the meeting.
Right.
There is a bar nearly at the bottom.
I don't have one.
There is a up arrow icon where you can share your content.
Right, open share tray, right?
Yeah.
Yeah, so that's not doing anything. Can you see anything from my side?
What can be done? Can you leave for a moment and rejoin? What's showing over there? అదే ఇప్పుడు
बिंदु संयुक्त नित्यं कामदं मोक्षदं ओंकाराय नमो नमः.
Good morning and a pleasant welcome to the fourth day of Biogen 2020. We are now moving to the welcome address. I now call upon Srimati Sujaya Lakshmi KV, Assistant Professor, Department of Biosciences, to deliver the welcome address. Over to you, ma'am.
Hello, thank you, Ashley. Good morning, everyone, and a warm welcome to the fourth day of International Webinar Series Biogen 2020. It's my pleasure to welcome all our participants to today's session. First of all, let me appreciate everyone for exploring this opportunity, joining the session organized by the Department of Biosciences, Sri Shankara College, Kaladi, particularly during the period of lockdown. Today, we have an eminent resource person, Dr. Padmapriya B Banada from New Jersey Medical School, Rutgers, USA, who will deliver a talk on the topic Innovations and Developments in Diagnostics in the Global Control of COVID-19 Pandemic. In the present scenario, today's talk becomes more relevant. Its significance is more pronounced, having known the role of diagnosis, monitoring, screening, and prognosis in fighting against diseases. Hence, I'm definitely sure that you will gain much from today's presentation. With extreme pleasure, I welcome Dr. Padmapriya for today's presentation. I extend a hearty welcome to all of you to the session and a nice time ahead. Thank you.
Ashley, please hold on, Ashley.
Yeah, thank you. I just need to make.
Excuse me, Priya.
Mm-hmm.
Are you okay? I mean, is it coming?
Can you see my screen?
Not yet.
Okay.
Just click on that share tray button.
Yes.
And wait for a moment.
Mm-hmm. Been waiting.
Sujaya Lakshmi?
Yeah.
Please off your video.
Okay, thank you.
Okay. Should open. How about now? Can't see my screen?
No, Priya.
Is there any other way of sharing this?
Okay, shall we do it from here?
Yes, sure.
If you could share that content via email, I can do it from my side.
Okay, it's a big file though.
Oh, is it so?
Okay, let me see. Let me send you a PDF file, okay? Did you get it?
Did you get? Let me check once.
came from my Gmail. So
Okay. Yeah, it has come. Let me download first.
sure.
Hello.
There may be some technical problem. We will be correcting it soon.
Uh, 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 Priya,
Yes.
actually, I have received it as a PDF file. Can you send the PowerPoint file?
It's a very big one. There are a lot of pictures. I don't think so.
Because
Email.
I'm not able to share the PDF in Teams.
Okay. Oh God. Why can't I?
Elisa, we saw the PDF. No, just now we saw the PDF.
Well, Samis, are you seeing the a complete slide of that page?
A complete slide means you will have to adjust the page. We saw the page. We saw the PDF, and I think if you adjust the screen, we will be able to see.
Can I share it on Google Drive?
Priya, just hold on. Our technical team is actually trying for that.
Dear participants, sorry for the inconvenience caused. Please stay with us for a while. We will be correcting it shortly.
Priya, is it visible now?
Yes, it is.
Okay, actually, I'll try to manage the content. Just please hold for a minute. Actually, we have few formalities to complete. Just 2 minutes.
I'm also trying to send you through Google Drive. Would that be okay? I'll send my slides.
If
Like to do that.
if it is coming, then we can proceed with this one. Otherwise, we'll go for that option.
Did you receive it?
Yeah.
Okay, let's try that.
Priya, it's a big file. Actually, it is downloading. Priya,
Yes.
shall we continue with the PDF file, which is present now, which you can see right now?
Right. You're not able to get that.
No,
far?
actually, it seems a big seems as a big file. It's downloading right now, and my network speed is not that much good.
Okay. All right.
Okay.
Yeah.
Just
yeah.
a minute. Yeah.
Mm-hmm.
Just a minute. Just 2 minutes. Hold on.
Sure.
Yeah. Ashley.
Yes, miss.
Yeah, you can proceed.
Okay, miss. Let's begin today's technical session. Today we have Dr. Padmapriya B Banada with us today to deliver the talk on current issues and challenges on the diagnosis of COVID-19. I invite Dr. Elisa PA, Assistant Professor of the Department of Biosciences, to introduce the resource person.
Okay, thank you, Ashley. First of all, sorry for the inconvenience caused. Hello, everyone. It's my privilege to introduce our today's speaker, Dr. Padmapriya B Banada. Dr. Priya obtained her doctoral degree from Mysore University, India. She has received research fellowship from Central Food Technological Research Institute, CFTRI, Mysore, and worked as a postdoctoral research associate at Bhunia Lab, Purdue University, West Lafayette. Later, she joined New Jersey Medical School as a research scientist, and she has more than 15 years of experience in molecular diagnostics of infectious diseases and tuberculosis. She is a recipient of the prestigious Purdue Agriculture Team Award and is also recognized for her outstanding service as online mentor for many years. She has received many research grants, including the grants from National Institute of Health and Department of Defense. Dr. Priya was a key player in the development of Expert MTB RIF assay and part of the team developing the Expert MTB Ultra. She developed the novel processing methods for blood and stool samples for the detection of TB from immunocompromised patients and children. She is now leading the projects on the development of advanced diagnostics in biodefense and sepsis, with a current focus on fungal sepsis and hemorrhagic fever viruses, including the Ebola virus. As of now, Dr. Priya has 5 US and European patents, product licenses to her credit. She has published over 49 peer-reviewed papers, several book chapters, and reviews. She worked in collaboration with FINT and CDRC on several clinical trials in Africa for the detection of TB. As per Scholar Metrics, Dr. Priya has nearly 4,000 citations, and she is also the reviewer of several journals such as Journal of Clinical Microbiology, Journal of Food Safety, BMC Medicine, PLOS One, all of which are high-impact journals. And she is also a member of many professional societies, including the American Society of Microbiologists, American Association for Academy of Sciences, National Food Science Association, etc. At present, she is working as assistant professor, Department of Medicine, New Jersey Medical School, Rutgers, USA. And today, Dr. Priya is here to deliver a talk on innovations and developments in diagnostics in the global control of COVID-19 pandemic. Thank you.
Thank you. Thank you for having me.
Thank you, ma'am. Moving on to the technical session. The session begins with the presentation of the speaker, followed by a Q&A session at the end. Dear participants, Please use the chat box facility to raise your queries if any. The posted queries will be posed to the speaker at the end of the session after screening. Once again, kindly make sure that your audio is muted and video is turned off to avoid any disturbances. Dr. Priya, now the platform is all yours.
Thank you very much. Thank you for that kind introduction. And it's my pleasure to be talking in front of all of you. And so I have been, as Dr. Anisa explained, that I have been involved in diagnostic development for a long time. And I have very honored to be part of COVID-19 development, diagnostic development as well. So I would like to walk you through. Anisa, I think there is something obstructing. Okay. Would you like to give me the control or? Oh. Hello, I don't see the presentation.
Yeah, it's coming, Priya.
All right. Were you able to download the PowerPoint? Meanwhile.
Priya, the PowerPoint is actually ready now. I'll share that one.
Mm-hmm. Yes.
Okay.
Yeah, I mean, I did send you the request, and should I?
Okay, okay. You do once more. I have clicked on allow.
Okay, awesome.
Is it possible from your side now?
I don't see the slides. Okay, I think better you control it. I think this is taking way too long.
Okay, okay. I'll do that. Yes, shall I move on to the second slide?
Yes, please. Yeah, sorry everyone. This has been the reality of the world right now, right? So all of us working from home, and technicality is always been a challenge. And so that's how we have wrapped ourselves. That's what my second slide explains. We are wrapped ourselves in a caution tape right now, and trying to stay away, be cautious, and the virus has taught us some different world order. And moving on to the next slide. I'm not going to go over too much about the virus itself, but let's understand what happened. How did we end up in this pandemic situation? So just to recap the timelines, it started kind of like as we know, it is December 28, 2019, WHO first announced the emergence of a novel coronavirus that has been infecting in China. And here in US, January 21st, CDC confirmed the first case of coronavirus. And in a week, they had the first test kit released. Then by February 11th, the name, which was used to be called nCoV, was changed to SARS-CoV-2 by ICTV. And on the same day, WHO announced the new name of the disease as COVID-19. And a month later, it declared COVID-19 as a global pandemic, considering the number of countries that were experiencing the disease. Moving on to the next slide. So immediately after COVID testing made headlines, everybody were like, oh, we can't find enough tests. And there were bigger headlines everywhere asking, oh, why are we lagging behind in testing? And so people were suspecting, okay, certain regions in the world is not experiencing anything. Is it because of low testing? So the COVID testing made headlines for all wrong reasons. Next slide, please. Before we move on to understand what COVID-19 diagnostics was all about, let's understand what is the importance of the laboratory diagnostics by itself. Diagnostics is not just about going to the doctor or going to a test, more wider applicability in case of a pandemic. So when a SARS-CoV infection happens, the testing starts with the diagnosis. So you need to have a test. Once it is diagnosed and tested and confirmed as a SARS-CoV infection, so it also helps further in doing an epidemiological surveillance. So once the infection is spread into a disease, and so various tests could be used to understand the stages of infection and also in monitoring the therapy and etc. And even after the person is recovered, there can be an epidemiological surveillance using antibody kind of assays where you can understand whether it is a long-term protection for the individual or is it short term. So there is a lot of advantages that diagnostics can offer to understand the complete picture of a pandemic, not just about going and getting tested. Next slide, please. There are several types of diagnostics for COVID-19. You may have heard about the Rdpcr-Based, which is pretty common. Then there is an antigen test. There are serological tests, and there are other developing methods, which I will be talking about in subsequent slides. These are the different areas I'll be covering in the next 45 minutes to an hour. And to start off with, let's understand what is a PCR versus serology, and why do we need two different tests. So as you can see in this graph, when the infection starts, the x-axis shows the timeline, the days of infection. So when you have exposed to a SARS-CoV infection in early on, like from -5 days to the start of the symptoms, the virus is replicating in the system. So that during that time, PCR becomes very, very useful. So in this graph, you see the viral load is by PCR. So you can continue to monitor the viral load in the patient until up to day 10. And then by about 5th to 6th day, the IgM, IgG antibodies kick in. And so once the antibody response is active, the viral load is seen to gradually go down. Next slide, please. So this is a study by Liu et al. In the European Journal of European Respiratory Journal, where the same data has been conclusively shown. They haven't taken samples before day 5, but you can see the trend. The PCR, the viral load has been, it decreases in about here in this case 15 days. and whereas the antibody response starts after about 15 days. So that's pretty conclusive. And so that is why the combination of a PCR test and an antibody test would give us proper monitoring for a prolonged period of time. Next slide. Okay, let's understand more about the Rdpcr-based molecular test. As I said before, it is used basically for the viral load detection in the early stages of the infection. So this cartoon kind of explains as an example of what happens in a molecular diagnostic test using the Rdpcr. So a patient's nasopharyngeal or oropharyngeal, any sample is taken, and once the sample is collected, either the whole sample is used in certain tests or an RNA is extracted and used in further analysis. I am presuming all of you are aware of RT-qPCR, but briefly, reverse transcriptase polymerase chain reaction happens when somebody needs to mute. Sorry. Can they please mute? Yeah, thank you.
Priya, unmute yourself. Priya?
No?
Okay. Unmute yourself. Okay, fine.
Can you hear me? Okay.
Yeah.
Let me go over this slide again. I don't know where I got cut off. So with the molecular diagnostic testing, once a patient sample is collected, the specimen is used as is, or it further goes into RNA extraction, depending on the test method that is being used. So once the RNA is extracted, it is basically almost all the methods used is either an isothermal or an RT-qPCR method, where a single-stranded RNA from the virus is reverse transcribed to cDNA and then further amplified by PCR. So depending on the assays, there are different, excuse me, can you stay in that slide? Thank you. So depending on the assay, there are different primers and different regions on the genome that are targeted, and the assay is developed based on that. And the test results are out in which shows whether if there is an amplification from a target gene, there is a positive curve, and a negative obviously will not show any amplification. So that is an overview. Next slide now, please. So this led to an enormous amount of testing kits to come to the market. So these are just few of them that I have put. So these are all FDA approved, US FDA approved tests. And can you click on your mouse? Can you click your mouse? There should be some. Okay, so these are the US FDA has approved about 161 different molecular tests, and there is like a like the flood of both antibody and molecular tests that have been coming to market now. And so, as I said, it's 161 molecular tests. There are 4 antigen tests and 45 serology tests as per the FDA website. Next slide, please. Next slide. Yeah, thank you. So why did we need so many tests in the first place? I mean, why couldn't have we managed? Because as I said before, if you remember, CDC came up with a test kit within a week of identifying the first infection in the United States. So what happened was the CDC test, somehow it yielded an unsatisfactory validation from different state and local public health labs here. And they later on figured it out to be a lab contamination of their negative controls. But they took a while to figure that out. And meanwhile, US FDA and the US government was becoming restless, and there was a lot of political and political pressure on the government to get the tests out. So what US FDA did was they just relaxed their rules to expedite the availability of diagnostics. Earlier, they had very stringent rules that that people had to abide by, and a lot of testing, a lot of validation that needed to be done before submitting or getting any FDA approval. But the rules were extremely relaxed, and so that kind of opened this floodgate. And so that has led to what we have 161 different tests, which is very good. That means a lot of people could get tested in the United States. Next slide. Next slide, please. So out of all these 161 molecular tests approved by FDA, there is only one point of care test, which is Cepheid's Expert Express SARS-CoV-2 assay. So next slide. So this test is, I mean, I'm personally very honored and to be a part of this test. And we were involved in the evaluation of this kit. And next slide, please. Yes. So, and we were able to get this very, very quickly within about 3 weeks. We were able to apply for the US FDA application. This was done in collaboration, and so I'm really very proud of this test kit. Next slide, please. I'm going to show some data that that's published as part of the US FDA and subsequently as a manuscript. Next slide, please. Yeah. So the test was basically designed to target the E and the N2 gene of the SARS-CoV genome. And next slide. I'm not going to go over too much on the genome sequence because I'm presuming all of you know much about it already from all these days of talks. But how does this express? It's very, very hands-free method. It is all it needs is the cartridge comes completely assembled, and it contains all the reagents that are required. So the sample is taken, basically 300 microliters of the sample is transferred onto the sample chamber in the cartridge and closed. And next slide. And then loaded depending on the kind of instrument the laboratory has. It could be any POC, small instrument to large scale infinity system. So what happens in the complete system is automated. So once the sample is added to the cartridge and loaded into the system, the nucleic acids are purified, and the purified nucleic acids mixed with the PCR reagents, and the simultaneous amplification detection happens, everything integrated in the cartridge, and results are ready to you within 45 minutes to 50 minutes. Next slide. This is an example of how the results look like if a positive SARS-CoV test. So you would see that the E and the N2 gene both would be positive, and along with an SPC. I'm sorry, the figures are not that clear in this, but the three curves that you faintly see represent the SPC, N2, and the E gene. And what if all the three are positive, it's considered a positive test. In a negative test, obviously, you will see only the internal control coming up and none of the target genes showing. And in case of an indeterminate, it's possible sometimes just the E gene shows up and not the N2. It's essential that both the N2 and the E gene are positive, because E gene has some conserved conserved region with other coronaviruses, and often it picks up. Not many, but and also it is more susceptible for mutations. So that is the reason the E gene is not used as a standalone target. So we were very happy to see the limit of detection of this express SARS-CoV test. We had a sensitivity as low as 0.01 PFU per ml, and which gave 100% positive rate, and very decent CT values. And in this test particularly, you see the CT values going up to 42.5, for 0.0001 PFU. In this particular test, any CT value is considered positive. There is no cutoff because our specificity was that good. Next slide. So when it was compared to other existing Rdpcr tests, it had a very good correlation, except for the Quest SARS-CoV Rdpcr test. So we did discrepant analysis in the next slide. So the discrepant analysis showed that actually when you do an additional testing, most of the samples matched with the additional testing rather than with the comparator test. So it's pretty good. It's, probably I think our test is much more sensitive. And as I said, specificity was very, very good, very decent. It was tested against a plethora of organisms, including all the human coronaviruses, SARS-CoV, MERS-CoV, bat-CoV, and all other common respiratory viruses and bacteria. So as part of the clinical evaluation, because this test was developed very early on, I think this was developed early in March, and we didn't have proper approvals to get the patient samples. And in the United States, in New Jersey, there wasn't many cases at that time. It had started in California at the time. So we had to do like a blind spiking study as a simulation for the clinical evaluation. So we did it at 2x, 3x, and 5x the LOD, and it correlated pretty well. So we were very excited. So not only from our hands as a completely as a third party, the FIND, which is a very popular name in among the diagnostic people, the Foundation for Innovative and New Diagnostic. It's a nonprofit organization, but it funds many diagnostic methods around the world. And so they did an evaluation study comparing different tests. And so they compared the two POC, near POC automated tests, Cepheid's Expert Express SARS-CoV-2 test, and India's MalBio TrueNat SARS-CoV-2 test. As you can see that Cepheid test holds pretty well. So with that, they also tested 21 other molecular tests in this round one evaluation. The sensitivity in their hands ranged anywhere from 90 to 100%, and specificity held very, very good. And the copy numbers, most of their tests were able to detect 1 to 10 copies per reaction. So in the next subsequent 3 slides, you will see more of these tests being done. And if anybody has any question, I can stop and take your questions.
It's okay, Priya. We have a question answer session at the end. We will deal at that time.
Sounds good. Sounds good. So you can move on. Next slide, please. So now, based on all this testing and the capability of and the reproducibility of the molecular testing is now considered the gold standard for COVID test. So everything was going good, but the major problem that we had was there was lack of swabs, and we weren't a lot of people weren't able to, the doctors and testing centers weren't able to get the basic swab and the transport media. And the nasopharyngeal swab itself was not a very pleasant experience for people to have. People started complaining that it's a lot of discomfort. They felt a lot of pain, and if the people were very sick, they outright refused to get the swab inside. So if you click on that, the swab, there is a small animation which shows that the green spot that is in the cartoon is basically the area of nasopharynx. So it has to go all the way up in order to get the sample. So it was not the best sampling site. And so that led to coming up with different sample types and different specimens to be used as an alternative method. Out of all these things, the oropharyngeal is something that was pretty commonly used, and so I have narrowed down for my subsequent talk based on the increased interest and the number of publications. Next slide. And focused all my efforts in this talk on saliva. So next slide, please. So saliva is something that is a very, very convincing sample, and people have an increased interest as an alternative specimen. It's easy to collect. Nobody complains much about spitting in a cup. And so there are a lot of publications that came in support of the saliva, and they showed that saliva's performance is equal to that of the nasopharyngeal or the oropharyngeal samples. So that led to creation of a lot of testing. So according to this bx.bio, they explain why we need this saliva testing. As I said, that it is much more easier, and it is also shown to be much more sensitive than the nasopharyngeal swabs. And since we had problem with the shortage of the swabs for sampling, and so this is going to be much easier, and going to be a right replacement for the nasopharyngeal. And this can be done at home by the healthcare worker themselves or by the patient themselves, and just get the sample. So it will reduce any cross contaminations or for putting the healthcare workers or professionals into risk. So, and also it doesn't need a lot of training because everybody knows how to spit. So I'm very proud to say that my university was involved in the first saliva testing for COVID-19. I was not part of this study, but I'm very proud of what we have accomplished, because right now this is the test that is sent to all the employees of Rutgers University before they can report back to work. And it's a very simple test. So basically there is a container, as you see in the picture, where you collect your saliva, and there is the blue liquid that you see is a preservative. And so once the sample is collected, you cap that onto the tube, and just shake it, mix it, ship it. And once it arrives in the lab, the RNA is extracted, and they use PE chemomagic automated specimen processing system, which is really cool. It can do a lot of samples at once, and they use the TACPath assay for running the Rdpcr in ABI's Quant Studio. So the results are within 48 to 72 hours, and this is proven to be very, very convenient, and it's widely used right now. So based on their FDA UAA filing, the test performance has shown to be pretty well. So this was, they did an LOD study using the copy numbers, and per ml of the sample, they were able to detect about 200 copies. conclusively. It's 100% positive, but they did not go below 200, so we do not have the data, but it looks to be very, very sensitive. Next slide. Here they compared head-on-head with the NP and the OP swabs. And they studied all their gene targets in the assay. As it's shown, there are 3 different gene target, NS and ORF1, and MS2 is the internal control. And it worked pretty well, and it was pretty correlative. So it could be a very good replacement for the NP swab. So when reporting for the patient, to interpret the results for the patient samples, if it is undetermined and only the internal control shows up, it's called negative. If any 2 out of the 3 target genes have a CT that is less than 37, it is called positive. If 1 out of the 3 is less than 37, it's indeterminate, so it will be asked for retest. And if everything is negative, then it is also invalid test, so it will be asked for retest. And so the New Jersey State Department of Health validated their method, and in their validation, there was 100% correlation. So this is another test that very recently got USFDA UA approval. This is called Saliva Direct and developed by Yale School of Public Health. And this is also very simple, straightforward test where a saliva sample is collected, a proteinase K sample is added to that, and after combining, it is added to a PCR plate and inactivate the PK at 95 and run the plate in the 1-step RT-qPCR. So, and this has shown a lot of interest, and this test, as a fun fact, this was used here for the NBA players when they had to go back to play, so they had to get screened. And so the NBA adapted this methodology to screen their players, and that was done every day. So that's pretty cool. And their data for this shows that it is very correlative with the Nasopharyngeal Swab specimen. And in the graph B to the left side, the positivity for the SARS-CoV-2 over the period of days of infection, they saw that actually saliva samples were better. They performed better in the early stages of infection compared to the later stages of infection. So I think anyways, PCR is used for detection in the early stages of infection, and this would be very apt. And on the right-hand panel, can you go back? On the right-hand panel, C and D graph, again, shows the correlation. And it is, since the days of the onset of symptoms, it's very clear that, depending on the patient, most of the positive rate weans off around 22 days. And that holds good whether it is saliva or nasopharyngeal swab. So it doesn't change. Okay, next slide. So there is a meta-analysis study which was done comparing all different studies that had used saliva as a sample, and this study has shown that he has studied about I think 56 different studies, and it has shown very promising data. Next slide. So this is the sensitivity of the different assays based on these 6 different studies, and it has shown about 91% sensitivity. And next, and about 98% specificity. So both are very promising. There are other saliva-based tests in the market, Diagnostic, which again has is a PCR-based test and shown that it works equal to other commercial tests. And then then there is University of Illinois saliva test, P23, etc. So what are the basic limitations of the PCR test? So it first and foremost is that it's good in the early stages of infection, but it cannot help us in determining true case counts and rates of asymptomatic infection, because everybody is, whether you're symptomatic, asymptomatic, it's going to be, if we are infected, it's going to be positive. And for many people, it seems to be expensive because there is always an initial investment on this instrument. If your lab already houses it, then it's easy. The test kits are not that expensive, but initial investment for a new lab would be difficult. And also for some of these tests, it needs higher technical skill. So as an alternative POC test, Antigen test has been very popular. So these are lateral flow kits, which are very much preferred by a lot of people. But US FDA did not receive many of these applications as such, and they received only 4, and the 4 have been approved, and so they are available. So basically, I mean, if I want to go a little quicker on this, the rapid antigen test is when the antibodies are bound to the paper, basically on the immobilized on the line there. And when the sample is added, the antigen basically goes and binds to the specific antibodies, which results in development of that defined colorimetric line, which indicates whether the test is positive or negative. Next. So moving on to the antibody-based test. So antibody-based are another is also called a serology test. It's basically a blood test that can detect if a person has antibodies to SARS-CoV-2. So unlike antigen test can be used on any specimen. Antibody needs blood because it's looking for the host antibodies. This can help identify people who may have been infected with COVID-2 or have recovered from the infection. And it can help in large seroprevalence studies, and it can help in characterizing transmission, identifying the true burden of the disease or the population-wide serum antibodies, and calculating pretty much accurate fatality rates. And it also helps define asymptomatic infections. And so far, US FDA has approved about 45 serology tests. Before we jump into the serological tests, I thought I'll give an overview of what a humoral immune response is. Most of you might already know this, just to brush up. So when a virus is infected, a person is infected with a virus, it stimulates, it goes and binds to the ACE receptors on the host cell, and the host then start the series of antigen-presenting cells, which leads to the formation of the plasma cells, which produces antibodies, which could be any. There are different kinds of antibodies, which goes and attacks the antigen and neutralizes it. That is in the best case scenario. So how does these serological assays work then? So there are different kinds of antibodies, as many of you know, and but diagnostically, the primary targets have been IgM, IgG, and IgA. The reason being, as depicted in the cartoon, it's during the infection. The IgM is expressed early on compared to IgG and IgA. So IgM is considered to be an indicator of an early infection, and IgG it persists for a longer period of time. So you can monitor whether the person is developing immunity towards this pathogen. Next slide. So this is also to briefly explain what a lateral flow assay strips are, and which are the near POC tests. And here, unlike the antigen test, which is very similar chemistry, instead of the antigen here, instead of the antibody in the antigen test here, the antigens are captured on the membrane. And when the blood containing the specific antibodies are added to the test strip, it gets absorbed, it binds with the antigen conjugate and travels through and binds with the respective antibodies and develops the color. So depending on where the reaction is, you can kind of interpret the result as whether it's negative or IgM is positive or IgG is positive or both are positive. So similar chemistry is used in ELISA-based tests. So it is just that ELISA-based tests are more of a liquid base than the solid base of the lateral flow. So US FDA approved serology tests. I'm focusing on one test here that is evaluated by the CDC and FDA, the Architect I-1000. This is targeting the IgG. So later on, I think the test initially was trying to measure both IgM and IgG titer, but when you look at the graphs here, the IgG titer goes down after about 1,000, whereas the IgG comes up. It is not very good at the low titers of the virus. So they combined both the test, and the test was pretty sensitive, up at about 90%, and the specificity was about 100%. So next one. The next slide, please. Okay. Can you move to the next slide?
Is it this one? Priya?
Yes.
Can you see the slide?
Yes, I do. Can you do slideshow for full screen?
Is it okay now?
I don't see full screen.
Priya, can you see it now? Is it slide mode?
No, I don't see it. Okay, now I do. Maybe I think there is a lag.
Okay.
Okay.
Is it okay
Yeah.
now?
Yes, I think you skipped a slide.
Okay.
Okay, perfect. Thank you. So there are 4 other antibody-based tests that is very, very promising. Beckman-Coulter's axis, and Select, Biorads, and Diacerins, which show better performance, better sensitivity than even the Abbott one. So that it's pretty exciting. And all these tests have been validated by FDA. And the next slide, there was a meta-analysis study done on a number of antibody tests that were in the market, and they, according to them, the ELISA-based and chemiluminescent-based. Next slide, please. Chemiluminescence-based assays were much superior to the lateral flow. Lateral flow was not a reliable test because there was an increased false negative rate. The sensitivity was pretty compromised in this study. So this paper is a very good paper. It's a very good review. That is a presentation by itself, and I'm glad that I was able to get a visual abstract of it. And this clearly shows here the ELISA had missed about 16% of, it yielded about 16% false results. And the worst was the lateral flow. So considering all these things, there is a lot of, although we like lateral flow assays, and because of its convenience and because of its point of care capability, but considering the limitations of it, and so we got to be a little careful when opting for that test. So there were another evaluation of a number of serological assays published in Nature, which compared the tests, and apologize for this clarity, but most of the tests were pretty decent. The overall sensitivity ranged anywhere from 80 to 90%, and some tests were better than the other. And overall, I think it is not, the study concluded it's not the target. I mean, it is not like RPD target was better, the spike protein was better target than the nucleocapsid or anything. It was the assay itself. And most of the ELISA assays or the chemiluminescent assays were performed much superior than any other. But the major limitation we have with the antibody assays is the specificity. As we saw that it can lead to either false positives or a lot of false negatives. And it's not a reliable test. And US FDA, in a very historic move, it removed 125 tests that were submitted for the US FDA clearance because they did not meet the required most minimal clinical performance needs. So at this point, I want everybody to think about what we are in and what we can do. So in Chinese, this is a very famous quote from John F. Kennedy, that the Chinese write 2 characters for the word crisis. They call it VG. V means danger, G means opportunity. So in crisis, we have to be aware of the danger, but we also have to recognize the opportunity. So some wonderful people have done just that. And that's what I want to talk next on, the new innovations that has happened. And the COVID, although it's been bad, but as a scientist, I think it's a wonderful time to be a scientist, wonderful time to be a student, science student, because we are learning so much during this course of pandemic. The next slides, in the next few slides, I will be talking about what are the new innovations in diagnostic that we have seen. This is an example of carbon nanotubes, which could be used as a as a sample processing delivery mechanism. And it has come up with a wave of innovative diagnostics now, and everything seems so fun and cool. So Harvard and MIT researchers are coming up with a face mask that lights up when it detects the coronavirus. So, I mean, not literally, but when it can be detected on a face mask. And there is a smartphone app that are developing, which would listen to your cough, the sound of the cough, because depending on, this is using an artificial intelligence, they are able to judge what your regular voice was and what if it is a dry cough versus a mucousy cough. And Israeli Defense Ministry has launched a voice test study already using this technology, and they call it vocal fingerprint. And there is another study where people are trying to come up with a handheld COVID-19 test that could deliver results within 5 minutes, which is similar to what you would see in a glucose monitor. So it just put a drop of blood, and it should be able to tell whether you have enough antibody response or not. And then there is sequencing, and also there is a CRISPR-based Sherlock system that is very, very exciting, that it got US FDA approval as well. So let me talk to you about the CRISPR-based technology. I'm sure many of you have heard about it. Was in news last was able to cut off part of the gene responsible for HIV infection that was present in both parents, and he in the infants, and was able to deliver the twin babies. And it became a very controversial subject, and but the technology offers a lot of advantages. So CRISPR's technology is in very, very briefly, it can cut a targeted region, and it can also help in detection. So in this system that they developed, so once a patient RNA is extracted, it undergoes an isothermal amplification, and the Cas12 protein, which can recognize a specific region, and it is bound with a guide RNA, which is viral specific, target specific. And can you please stay on that slide? Can you go back and stay on that slide, please? Thank you. Yeah. So, and it uses similar to the lateral flow principle. It can capture the amplified RNA on a colorimetric method. And this is very, very sensitive. It can detect about 10 copies per microliter. And as the data has shown, because it's isothermal, with the increasing copy numbers, you do not see the effect of dilution. And so basically, if there is a reporter molecule that is present along with the amplified viral RNA, and once the RNA binds to the guide RNA, the reporter RNAs are chewed or cut at specific regions by the CRISPR protein. And once it is cut, this is dark because of FRET activity. And once it is cut, the fluorescence is released, and basically the color could be detected either in a solution form or on a strip. Okay, next slide, please. So this is a publication from the team that developed the Sherlock Diagnostics. And so in this testing, they tested different NP swabs from the patients, and they were pretty successful. And initially, the way they did it was they took the swab or the saliva and transferred it to the Sherlock mixture, master mix, and they did the isothermal amplification, and they dipped the strips. And in future, this is their vision on the bottom right. So they want to integrate into a cassette, which is under development, which can do isothermal amplification in a cassette, and probably transfer the data after reading the strip to your smartphone. So it can, so they're envisioning it to be done in-house by individuals, and it doesn't need a laboratory to run these kind of diagnostics. Next slide. Yeah, so this is my, I think, last slide, almost reaching the last. So the sequencing has been extremely, I mean, it is advanced tremendously in the past 10 years. So Illumina has come up with a next generation sequencing based in vitro diagnostic test. It's still cumbersome. It is not as easy as it sounds. This still requires a lot of workflow management, and the instruments are big, and it could be done in a centralized facility with proper expertise. But it offers enormous advantage, because here they have developed the test where you could you could target like 98 different amplicons that can amplify on the SARS-CoV genome, and there are 11 human internal controls that could be amplified, which makes it very, very clear and sure. And also the data output can also help you analyze if there are any mutations that are emerging in different strains regionally or in a particular population. So very exciting technologies so far, and this has given a lot of opportunities to learn. And the more we absorb these things, I think it's really cool for us. Next slide, please. Almost done. Next slide. And also all this information, they promise to be processed and get it out within 12 hours. And they can process up to like 1,500 to 3,000 samples at once. So it's a pretty high throughput method, which can offer a lot of advantages. Are the slides frozen? Okay. So that's I owe to my colleagues who have been very, very instrumental in getting the COVID test done, and that is us in the BSL-3 suits. That's, we call it space suits, where all the SARS-CoV-2 RNA work was done. And that's the center that I work in. And that was when the SARS-CoV test was released. And we were very excited to be testing in the laboratory. And with this, I take any questions you may have. Thank you so much.
Thank you, Dr. Priya, for your very scholarly and informative presentation. Now the session is open for Q&A. Participants, please put your questions and queries in the chat box. There are no queries yet. Can we please check the YouTube chat box too, Anisa ma'am?
Actually, there is a question in the chat box of Teams.
Okay, I read it. In case of viral antigenic drift or a shift, does molecular methods are useful? It can be useful. Well, depends on the test, but most of the tests are going to detect it as positive, negative. Whether it depends on whether it is probe-based. So most of the tests they don't fail with a SNP. So there can be still a positive reaction, but it depends on how many mutations have been acquired in that region, in that particular gene of target. That's why if you saw carefully, most of the tests are not developed for a single target. They have chosen multiple targets to circumvent this kind of a scenario. Okay, so what are the challenges faced by the labs in case of testing the samples for COVID-19 diagnosis? That's a good question, actually, because we do face those kind of challenges. The first and foremost, like in the beginning of this, I mean, I can answer that in two different perspectives, right? Like the supplies wise, there were a lot of dearth of PPEs. There was, as I was saying, there were a lack of swabs, and the basic materials was lacking. So that could be very challenging. And in the lab itself, when we started, we didn't know much about this virus. So lab safety-wise, personal safety, biosafety-wise, we didn't know a lot about how safe is it to handle these samples. But we in Rutgers, we employed the highest possible safety precautions, and we always made sure mask was always on, even before masks were made mandatory. And so, yeah, there are pretty good challenges that labs face. Does that answer your questions?
Priya,
Yes.
can I ask a question?
Sure.
Okay, Priya, what about these diagnostics are how much feasible for a developing country like India?
It is pretty feasible. The TrueNat MolBio tests are seriously very good tests. So we have had the opportunity to evaluate those tests for mycobacterium in our lab. And so it's pretty feasible. It could be done. It is, I don't think so it's super expensive. And it should be. I don't see a reason, because India is much more prosperous and developed, in the verge of development. It is not an underdeveloped country anymore. So I think a lot of very good technologies are coming from India itself. And it is just that we need a proper regulatory body there, which can, just like our FDA or CDC, I know ICMR is doing a very good job during the season and regulating a lot of these kits. And having a proper regulatory body would also help India get better, cheaper, more feasible tests.
Just a minute. Can I check once more in the YouTube channel, because the session is on live right now?
Mm-hmm. Sure. It feels really good listening to the cock sound. I missed that from India.
I think so far no questions, Priya.
Awesome.
Oh, okay, one question is there. Shall I ask?
Sure.
That is from Professor Muhammad Hatta Abdullah. Why there is a difference in the sensitivity of ELISA on COVID positive and negative?
Can you repeat that question?
Why there is a difference in the sensitivity of ELISA in COVID positive and negative results, maybe?
I mean, the COVID, I'm not sure if I understand the question properly, but the ELISA test for the COVID positives defines what is the sensitivity of the assay, and ELISA on the negatives defines how good the specificity of the assay is. So in general, antibodies are, depending on the assay that is developed and how good an antibody that was produced, there can be cross reactions with other inherent ones. So that's why the negative samples are high and the sensitivity compromises because of that. So that is why it gives a false positive results at times because of not a very good antibody. But there are in this pool of samples, in this pool of tests that they have developed, there are very good tests which have shown very high specificity, but compromised sensitivity. Does that answer the question, or?
I hope so, Priya.
Okay. All right.
Okay. One more question is there in the teams from Adira Rajesh. China has recently celebrated their success against COVID. What did they do differently as compared to other countries?
Politically, I cannot answer that question. I wish I knew too, because US is still not able to come out of it. So we really don't know, and there is no publication. I'm a scientist. I like to go by science and not politics. And I haven't seen any publications that China has produced, and there is no information on mass vaccination or any of those kind of mitigation efforts. And I can't, I'm sorry, I can't speak too much about that.
Okay. That's all about our QA session. Ashley, you can proceed with the rest.
Okay, Miss. I believe that our speaker has cleared almost all the queries. Dr. Priya, would you like to share anything more?
No, I think if I was able to convince all my listeners and on the importance of the diagnostic test, then I have done my job well.
Okay, thank you. Dear attendees, now the feedback link is available in the chat box, so participants make sure that it is noted. It's time to wind up our session. I invite Srimati Priya Krishna, Assistant Professor of the Department of Biosciences, to deliver the vote of thanks. Ma'am?
Thank you, Ashley. Thank you to Dr. Padmapriya B Banada for your informative talk on the topic Innovations and Developments in Diagnostics in the Global Control of COVID-19 Pandemic. How all of you have done well through the webinar. It gives me an immense pleasure and privilege to propose the vote of thanks to this event. The current COVID-19 pandemic is unprecedented. The scientists and global health professionals accelerate the research and development process in order to diagnose and to find therapeutics for this novel coronavirus. Today's talk has given us insight into the current level of knowledge about the new strategies employed and the challenges faced in the field of diagnostics of an outbreak like COVID-19. Hope it really aligns of the talk was really interesting and meaningful for all of us. I would like to pay my deep respect to all the participants for your positive participation in this webinar. So, on the behalf of all the participants, organizers, and institutions, I convey our profound gratitude to Dr. Padmapriya
for raising your important work and sharing with us your findings and opinions today. I conclude that the purpose of the webinar has been completely accomplished. Once again, I thank all for your cooperation. Thank you.
Thank you very much.
Thank you, ma'am. Our last technical session of this webinar series is on 22nd September 2020. The link for that will be sent to your email ID before the start of the session. Once again, I thank everyone for your kind cooperation. Dear participants, we once again remind you all to fill up the feedback form as early as possible before leaving. Active only for 1 hour after the session. It's the time to disperse. See you on 22nd of September 2020. Thank you all and have a great day ahead.